You're listening to Conferences Online Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is March 16, 2012, and I'm your host, Dr. Jay Portnoy. Our topic today, cell function assays. Our presenter is Dr. Marcia Chan. She's an investigator in immunology research at Children's Mercy Hospitals and Clinics. Our first speaker this morning is Dr. Marcia Chan. Dr. Chan is an investigator in the Department of Immunology Research here at Children's Mercy Hospitals and Clinics, and she's agreed to update us this morning on cell functional assays. So, Dr. Chan, welcome to Conferences Online Allergy. Let me just get the slides up. I think that's it.
Okay.
And
Great.
Take it away, Dr. Chan.
Thank you. Okay.
Yep.
So I guess we'll start with our internal CME credit. The first question is, which of the following assays could be used to measure proliferation of T cells? CFSE, chromium release, tritiated thymine uptake, A and C, or B and C. Okay. The second question is, which of the following objectives of cell culture does not directly represent a present or planned therapeutic strategy for addressing human disease? TILs, differentiated stem cells, cytotoxicity testing, or monoclonal antibodies from Hybridoma. Okay. And the third question is, measurement of antibodies by ELISA is useful as an indicator of immunodeficiency, multiple myeloma, autoimmunity, or all of the above. Okay. So we'll review those questions after the talk, and hopefully your answers will be the same or different. So today I want to talk about tissue culture, and that's the growth of cells in vitro. We'll discuss the various culture conditions, the patterns of growth, and how one goes about counting cells. And then we'll specifically talk about the isolation of immunologically relevant cells, and then go through some of the assays for lymphocyte function. So tissue culture is really the growth of cells, tissues, or organs in an artificial environment, and that environment consists of a culture vessel containing nutrients that are essential for survival and growth. And we can do organ cultures, and that would be whole organs or intact organ fragments, or cell culture where we've isolated individual cells from those organs. So when we do tissue culture, there are basically two, forms of cultures. There are the monolayer cultures, and these are usually epithelial-like or fibroblast-like phenotypes. And basically, they grow in sheets on the bottom surface of the tissue culture flask, and they grow until what is referred to as contact inhibition, unless they're transformed. And so on here on the left are epithelial cells growing as a sheet on the surface of the tissue culture flask, and you see the epithelial cells have a kind of a boxy type phenotype, and they grow as a sheet, and they grow until they hit their neighbor, and then they stop growing. That's what's called contact inhibition. And here's an example of fibroblasts growing, and you can see that their morphology is quite different, and they're very spindle-like. And again, as soon as they meet their neighbor, then they will stop growing unless they're transformed. And if they're transformed, then what you see is they start growing on top of each other, and you can actually see kind of a three-dimensional shape to the culture, to the tissue culture flask growth. And then we have those cells that grow in suspension, and these cells grow, they float free in suspension. For example, lymphocytes do that, and when you look at the cells under a light microscope, what you'll see is that you'll see them in various dimensions within the media. So you'll see some that are farther down, and you'll see some that are close up. And then these are just two forms, two pictures, one with less cells and one with more cells.
Is there a maximum size that the clumps can reach before the nutrients can't get to the new cells?
No. So when they're in suspension, they will continue to grow and grow, and then they'll finally deplete the media, and then when they start to deplete the media, then they'll start to die.
The cells in the center of the clump, how can the media get from the outside into those center cells? It can diffuse all the way?
Really? Yeah.
Really?
Yeah. You can. And these are usually floating, so they'll probably get media from the bottom.
Mm-hmm.
Right. Okay. So the culture conditions, you have to have the appropriate temperature, and most likely that would be body temperature of the host. And so for mammalian cells, we grow them at 36 to 37 degrees Celsius. For insect cells, they grow at 28, and chicken cells grow at 39. If you're looking at cells that grow in a monolayer, then you want a good substrate for attachment. And so you basically want to mimic the extracellular matrix, so that you might coat that vessel with collagen, gelatin, or fibronectin. You need an appropriate pH, which is physiological, which is in the range of 7 to 7.4. And then the cells need essential gases. And most cells, human cells, need 5% CO2, and stem cells need a little bit, also need 3% oxygen. Now, the culture media is made up of both defined ingredients and undefined ingredients. So the defined ingredients consist of amino acids, vitamins, minerals. We need antibiotics in there, such as penicillin and streptomycin, and then something to control the pH, so usually a buffer such as HEPs. Because as the cells grow, they'll create acidity in the media, so you need some sort of buffer so that the media isn't always really acidic. And then there are those ingredients that are what we call undefined, and this is provided through animal sera, and they provide the growth factors and hormones that your cells need. Okay, and so here's just a list, and this is by far not a complete list of the types of media that one can use to grow these cells in. And the ones that are highlighted in red are the most common ones that we use for lymphocyte growth. So there's the Delvecco's modified Eagles media and RPMI 1640. And if you compare those two types of media, this is pretty small, but what the asterisks indicate are the differences between the two types. And so you see kind of right off that the RPMI 1640 has more amino acids than the DMEM and a few more vitamins. And then with the DMEM, it has actually a higher concentration of glucose than the RPMI. So depending what your cell's needs are, you might grow them on either RPMI or DMEM. Okay. So now, to determine whether or not your cells are growing successfully, you can look at their morphology. So if you start to see changes in the shape of the cells, or if the cells, say, grow as a monolayer and you see that they become detached, or you see that the cells are granular, that's not a good sign. That means the cells are in stress. You can also look at their growth rate. So if the cells are growing happily, you know that the cells are doing well. And so you can measure growth rate just microscopically or by counting them. And so you see here is a microscope view of cells, lymphocytes at day 1 and at day 7. So you see that by day 7 there are a lot more cells than at day 1. So that's an indication that they're growing fine in the tissue culture. So if we wanted to enumerate cells, we can do them in a couple different ways. We can, if you have the advantage of having a culture counter, you can use that. And what that does, it detects the change in the electrical conductance. And so resistance change is recorded as an electric current, which is then correlated to particle size. And so here's the probe that is for the culture counter, and then you would put your cells in this cuvette here, and then the number of cells would be read up here on the screen here. The old-fashioned way of counting cells is using a hemocytometer. And so this is a specialized microscope slide, and it has these chambers here in the center, and there's this little groove here where you introduce your sample. And then when you look through the microscope, you see this grid pattern. And normally what we do is we count the cells in area 1, 2, 3, and 4, and then take the average, and then that is the number of cells you have. And because this is a 1.1 millimeter depth sample, then that count is times 10 to the 3 and whatever your dilution factor is. Now, when you look at the grid here and you see your cells, you want to be able to count the cells. If you have too many cells, then you're not going to get an accurate count, and so you'll need to dilute the cell suspension. And so this view here is a pretty good view of the correct dilution and their accuracy in which you can count the cells. And so in a particular grid, if you can count 100 cells or less, then that's probably a good range of dilution that you've made for the cells. Okay, now obviously when you're handling cells in vitro, you're going to have contamination. And there are 3 basic forms of contamination that are associated with cells. So there's bacteria. And if you get bacterial contamination, you can kind of see the bacteria. It's kind of got, they're really small spots. They almost look like kind of brown, they do this browning motion. And so, and the cells don't look well. Okay. If you have yeast contamination, you can actually see the yeast, and they're going to look different than the cell. So here's a normal cell, and here's yeast. And often when you open up your tissue culture flask, you can really smell the yeast. So that's pretty good indication you've got yeast contamination. And then if you have fungal contamination, you can see that quite clearly as well.
Can it be contaminated by other cells too?
Hopefully not. If you do your tissue culture correctly, then you should not have contamination of other cells.
Because I just remember people who work with like HeLa cells used to say they get into everything and they contaminate it.
Yeah, well, that's your tissue culture.
Okay, it's got to be good technique.
You have to have good technique. Okay, so what can we use cell culture for? We obviously for virology, for growth of viruses, for the study of infectious cycle or clinical detection and isolation of viruses. Cell-based manufacturing for vaccine production, for polio, rabies, and measles, they're using cell culture. Also monoclonal antibodies from hybridomas. For gene therapy, to replace missing or non-functional genes. For cell therapy, and here we grow tumor-infiltrating lymphocytes, or we can differentiate stem cells, and also for toxicity testing or drug screening. That's what we use in vitro cultures for. Okay, so let's turn our attention now to the immunological in vitro cell culture systems. We can have primary lymphoid cell cultures, and so this is where we would isolate lymphocytes from blood, lymph nodes, thymus, or spleen. This would be a heterogeneous population of cells, and their propagation is really only for a limited time. You can keep them growing for quite some time if you add maybe a cocktail of cytokines, but they don't have a kind of an unlimited length of time in which they'll grow in vitro. The other way is to clone lymphoid cell lines, and these are essentially tumor cells. And you can do this also by taking a normal cell that has undergone transformation, either chemically or by a viral infection. And obviously a clone lymphoid cell line is a homogeneous population. Every cell is the same. And these cells have an infinite lifespan. And then also, obviously, we also have hybridomas as well. Okay, so if we wanted to generate a B cell clone, for instance, we can take a population of B cells from, say, peripheral blood or a lymph node. We can infect those cells with Epstein-Barr virus. That basically immortalizes the cells. And then by what is referred to as limiting dilution screening, we can isolate these cells so that we have one cell per well. Then we can screen the amount of, or the type of isotype, the antibody that each well is secreting, and then say if we're interested in IgA or an IgG or an IgM secreting antibody, then we can take this cell and then start to propagate it, and then we have what is referred to as a cloned B cell line. To do that with the T cell, it's a little more challenging in that we have to stimulate the T cell after we've isolated with antigen and antigen-presenting cells for 4 to 5 days. And then we rest those cells where we still need a feeder cell layer, but there's no antigen. And then we do a restimulation. And then we would do the same thing that we've done with the B cell, and we would clone by limiting dilution. Again, we need to have a feeder cell layer for the T cell, plus antigen, plus IL-2. Okay, and then we can generate a T cell clone that way. Now with the hybridoma, and I think Dr. Barnes has probably gone through this with you, or will go through this with you, Kohler and Milstein won the Nobel Prize for the development of the hybridoma. And this is kind of an oversimplification of how one develops the hybridoma. But basically you take, you immunize a mouse with your specific antigen. Then you isolate splenic B cells from that mouse. You fuse them with a specialized myeloma cell line that doesn't secrete antibody. And you fuse these two together and you create a hybridoma. And then by a specific screening method and particular media, you then can do an ELISA to determine the specific antibody of interest that you want. And then you can generate that particular hybridoma. And these are now monoclonal antibodies, and then we can use them as therapeutic antibodies. Okay. So the cell types for the lymphoid cultures, we can have mononuclear cells, which include lymphocytes and monocytes, or we can actually isolate T cells and B cells. So if we wanted just to look at white blood cells, we just wanted to isolate those, we can do that simply by centrifugation. So we would take peripheral blood and spin it, and what you would see is you would have plasma on top, and you'd have this buffy coat layer. And what that buffy coat is the white blood cells and platelets, and then you'd have the erythrocytes on the bottom. If you wanted more specific cells than just white blood cells, if you wanted mononuclear cells, and then you would do what is referred to as a Fecal-Hyphae density gradient centrifugation. Okay? And so you again start with peripheral blood, but now you put it on Fecal, and at the top you would have plasma, and then you'd have this layer, and this would be your mononuclear cell layer. So that would be lymphocytes and monocytes. And then you'd have a Fecal layer here, and in the bottom here would be the red cells and your polymorphonuclear cells. Now, if you wanted to isolate the lymphocytes from the mononuclear cells, first you would need to deplete the cells of monocytes. And the easiest way to do that is by adherence to plastic. And 40% of monocytes in the blood, I believe, are 40% of the mononuclear cells in blood are monocytes. And so we could just take those mononuclear cells from the post-Fecal and just put them on plastic and incubate them at 37 for a couple hours. And then just gently take off those non-adhering cells, which would be the lymphocytes. If you wanted to isolate T cells, then you can do so by forming rosettes with sheep red blood cells. And on the surface of the T cell is a protein called CD2, and sheep red blood cells bind to CD2. And so once you form these rosettes, then you can take that combination of depleted monocyte layer, which would be the T and B cells, put it through another Fecal layer, and what you would have is on this layer, on the top would be the B cells, and the T cells, now because they're heavier with these rosettes, would be on the bottom. And so you could take your B cell layer off, take the Fecal layer, and then you have your T cells, and then you could just lyse the red blood cells, and then you'd have your T cells. Okay. If you want to do it more in a fancier method, you can do it by magnetic separation. And so what you would do is you would bind an antibody to the cell surface protein that's linked to a magnetic bead. And so say if you wanted to look for a CD4 population in your lymphocyte population, you'd have bind an antibody that's to CD4 and that's linked to a magnetic bead. You'd add this antibody to your population of lymphocytes, and then you'd put them in this column with this, fitted with this magnet. You'd put your cells through here, and those cells that bound to the antibody would stay in the column here. The other cells would be flushed through, and then you remove the column here from the magnet and then elute the cells that you retained here, and then theoretically that should be your pure population of, say, CD4 cells. So you can do it that way as well. And you can combine this too. So you could do the rosette first to get your T cell population. Once you have your T cell population, then you can do the magnetic separation to get, say, your CD4 population out. Okay, so assays for lymphocyte function. There are proliferative responses you can look for, cytotoxic activity, which then would be limited to T cells, and antibody production to B cells. So if we were to look at the proliferative response, you can do one that would be an assessment of the overall immunological competence of T or B cells. For instance, can the cells respond to a proliferation signal? Or if you wanted to look at an assessment of an individual's response to specific antigens, a low response may be indicative of a general or specific immunologic defect, or you can also look for HLA or MHC compatibility with a proliferative assay. Okay, so the types of proliferative assays, the most common one is the mitogen-induced proliferation. And for T cells, they use phytohemagglutinin, or PHA, and this is used clinically to test for immunocompetence. For B cells, they'll use Staphylococcus aureus, and sometimes for either T or B cells, they'll both respond to forbol esters and ionomycin. You can also do a one-way mixed lymphocyte reaction, which is called an MLR, and this will test the lymphocyte response to stimulation by irradiated or mitomycin C-treated allergenic lymphocytes. You can do an antigen-induced T cell proliferation, and so you could test the proliferation of T cells in response to a specific antigen, such as tetanus toxoid or allergen. And then there's also a lymphokine-dependent cell proliferation, and here you would test the dependent nature of a cell population for a specific cytokine. Okay, so how do we measure proliferation? We talked about measuring them just numerically using a hemocitometer or Coulter counter. We can also score for DNA synthesis using tritiated thymidine or BRDU. We can score from healthy mitochondria using MTT or by counting cell division by CSSC. So let's look at the thymidine and BRDU assays and looking for DNA synthesis. So looking at thymidine, thymidine is incorporated into DNA as the cells divide. So we can collect the cells onto filter paper discs and count them in a beta counter. So we would expose the cells to the mitogen and then add thymidine for, say, 16 hours, and then we'd harvest the cells. And so this is what we used to use. It's called a PhD cell harvester, and this is what we would do. And here there are spigots here that you would aspirate the cells, and that would go onto this filter paper. And then those filter papers would be cut into discs with this contraption, and they would be placed into scintillation tubes, and then you would fill it with scintillation fluid, and then you count those in a beta counter. And the results would be, say, you would have cells that have been untreated, so they're not treated, they're just media alone, and then with PHA, and then this would be your PHA response. And so that would be in counts per minute. And so depending on how great your lymphocyte response is, you can have, like you could maybe 10,000 or 35,000, depending on how well your lymphocytes responded, or your T cells specifically responded to PHA. You can also do it by bromodeoxyuridine. It's similar to the DNA precursor thymidine, and the cells incorporate BRDU during the S phase. And to assess those cells, then you need to stain with an antibody to BRDU and then analyze this by flow cytometry. So here's an example where the cells were stained with both CD4 and then an antibody to BRDU. And here are the cells that were non-stimulated, and you can't even actually see it, but 0.2% of the cells are positive for BRDU. And so here, these are the CD4 cells that they exposed to whatever antigen. And so in this case, you see that within the presence of antigen, 20% of those CD4 positive cells did respond to the antigen. Okay. and that's depicted in this quadrant here. Okay, so we can also measure proliferation by scoring for healthy mitochondria using an MTT assay. And this MTT stands for this big compound here. And the MTT is reduced to an insoluble purple formazine by mitochondrial reductase, and then that formazine is solubilized, and absorbance is read in a spectrophotometer, and the absorbance is directly related to the number of viable cells. And so here's just the spectrophotometer here, and this is an MTT. And so here's an example of an MTT assay where we've looked at absorbance at 540 nanometers. Here's non-stimulated cells, and here's the cells stimulated with PHA, and here's the patient cells that have a lower response to PHA than the patient, or the patient cells with a lower PHA response than the normal. The only, I think, drawback to using an MTT as opposed to, say, uptake of thymidine is that your sensitivity, I don't feel, is as good because the spectrophotometer only has a value of up to, say, 4, so it will only read up. So you can only measure so much absorbance, and then after that it's just too much. And so, whereas with thymidine you can, go up to as much as the counts allow you to do. So, but in those cases where you don't have the ability to do radioactivity, then this is probably, or the BRDU might be your best choice. You can also count cell divisions, and this is also by CFSC. And the CFSC is this ester, and it undergoes esterase cleavage upon entering the cells, and it diffuses throughout the cytoplasm. And so as the cells divide, the dye is split equally between the daughter cells. And then the signal diminishes with each cell division. So you can monitor the kinetics of the cell cycle via flow cytometry. So here's an example of T-cell proliferation. And so here is a case where the cells have not been stimulated. So the CFSC is going to be very bright, and so the peak goes way over here. Okay? And here is an example of T cells that have been stimulated with PHA for 3 days. And what you can see, and so this is the gate of the non-stimulated cells. So what you can see is that 50% of those cells have responded to PHA, and that they have divided. And you can actually count the number of divisions. So here we have 1, 2, 3, 4, 5. So there have been 5 divisions over 3 days in which the cells have responded to PHA. Okay? So this is with CFSC. And here's another example. And say if you wanted to look at CD4, so you stain the cells with CFSC, and then you stimulate them, and you also stain them with CD4. Okay? And so you see that the cells have been stimulated with PHA. You look at the CFSC response, and you see that there are, looks like 1, 2, 3, 4, maybe 5, but definitely 4 populations of CD4 positive cells that have responded with PHA. And these cells are likely the CD8 that have responded, but they're in this quadrant here, so you know that they're not CD4 positive. The CD4 positive cells are here. And so these are the other cells that are CD4 negative that have also responded to PHA. Okay? And here's just an example of Tregs cell proliferation, and this is with, so to look at Tregs, you stain with FOXP3. And this is staining T cells with different stimuli, antibody to CD3, or antibody to CD3 or ICAM1, or CD3 and CD28, or ICAM alone. And you see that with stimulating the cells with antibody to CD3 and ICAM1 is the best combination to get Tregs, and that's shown up here. Okay. You see that CD3 and CD28, you get a lot of proliferation, but those are not Tregs. Okay. So you can do a lot of different things with CFSC and a specific target population you want to look at. Okay. All right, so my interim summary here is measurements of proliferation. You can count the cells, you can score for DNA synthesis, or you can look at for helping mitochondria, or you can count the actual cell divisions. Okay, so the next one is to look at cytotoxic activity, and this would be the CD8 positive T cells. And so the indicators of T cell effectors, which are the CD8 population, you can either do polyclonal activation or you can do an antigen-specific activation. And the assay would be to look at cytotoxic effector cells, and then you need a target cell population. Okay. And this is just depicting what a cytotoxicity assay would look like. So you have your CD8, your CD recognition of the target cell through the T cell receptor, and this is just a scanning microscope slide of the CTL cell going towards the target cell. Then you have a conjugate formation after you have the recognition of the T cell receptor. Okay, and that's what this is here. And then once you have that conjugate formation, then you have cytoplasmic rearrangement of the granules within the cytotoxic T cells. So they all migrate to this area here, and that's what's happening here. And then you will get granule release through porphyrins and gran enzymes. And then you get the granules will then basically go through here, and they'll kill that target cell through apoptosis, and then that CTL will be recycled and go looking for another target cell. So the assay, there are actually 3 different assays you can look for cytotoxic activity. The traditional one is a chromium release assay, which is a radioactive assay, where the target cells are labeled with chromium. There's a colorimetric assay in which we measure lactate dehydrogenase, and that enzyme is released from damaged cells. And then there's a chemiluminescence one, which looks at adenylyte kinase, and this is also an enzyme that's released from the damaged cells. So if we look at the classical chromium release assay, we first label the cells, the tumor cells, the target cells with chromium, and then we add the target cells at specific ratios, and then we determine, then we collect this supernatant in these cultures and look for chromium, because the damaged cells will release chromium, and that's an indicator of cytotoxic activity. And then you can graph this as a percent cytolytic activity versus the effector target ratio. And so here is the control where you have no effectors, and sample 2 is you have a specific effector, and then here's another one with another effector. So the best killers show the lowest effector target ratio, so you see that sample 1 is your best population of killers compared to sample 2, because they kill to a greater extent at a lower effector to target ratio. Okay. So the lactate dehydrogenase assay, the lymphocytes and target cells are added together, again at a specific ratio, and then you collect the culture supernatant and you assay for lactate dehydrogenase, and the amount of color is proportional to the number of dead cells. And so this is just the assay that we use for that. And if you want to graph that, this is an absorbance assay, so you look for OD values, and so here this is what the results would look like. Here is the CTLs alone, and then this is the target cells, and this is what it would look like with a combination of them together, a different killer to target ratio. Okay, now the third assay for lymphocyte function is an antibody production, and of course we would be looking at B cells now. And an indicator of B cell differentiation, we can look for antibody production for immunodeficiency, autoimmunity, or cancer, as in the case of, say, multiple myeloma. Okay, and so if we wanted to look at antibody production, we could stimulate the B cells with different antibodies to these different surface proteins that the B cells express. So say if we wanted to look at IgE, we would stimulate the cells with an antibody to CD40, and we would add interleukin-4, and that drives the B cell to produce IgE. If we wanted just to look at a polyclonal activation, so we're looking at all B cells, then we might use Staphylococcus aureus. Or say if we wanted to look at IgA, for instance, then we might use interleukin-5. So we would culture the cells for 7 to 14 days after we stimulated the cells, and then we would harvest the culture supernatant fluid, and then we would look for the specific antibody that we wanted. So if we had stimulated them with antibody to anti-CD40-4, then we would look for IgE, or if we were looking for polyclonal, then we'd probably look for an IgM. And the way we would do that is through an ELISA assay, this enzyme-linked immunosorbent assay. So we would take the cell culture supernatant fluid. And so that assay, so on a 96-well plate here, we'd have the capture antibody. Then we would put our cell culture supernatant fluid on here, which would contain our antibody. Then we would have our detection antibody next. So say if we were looking at IgM, this would be anti-IgM. We would have IgM here, and the detection antibody would be, again, anti-IgM. And then we would maybe have a secondary antibody here, which would be a different antibody. So this would be an anti-IgM, and it would be made in a different host. So if this is human, this would be, say, goat anti-IgM, and this is linked to HRP, horseradish peroxidase. And then we would add the substrate to the peroxidase, and then the amount of IgM, say, that's in this culture supernatant would be proportional to the color change. Okay, so here's an example of what an ELISA plate would look like, and the more intense the color is, the more antibody you have in that culture supernatant fluid. Okay. The other way that you can do that, say, is by what's called an L-spot assay, and in this case, we're measuring actually the number of immunoglobulin secreting cells in the culture. So this is a little different from the ELISA. Again, you would have the capture antibody on the surface, and this would, now we probably use a tissue culture plate. And now instead of putting down the culture supernatant, we'd actually put the immunoglobulin secreting cells on this plate. Let them go for a little while, and they're going to be secreting antibody. So that antibody would be bound by that captured antibody that's on the plate. And then again, we would add our conjugated detection antibody, and then we'd add our substrate, and then we would do a series of washing, and then we would develop it. And now what you would see is every single one of these purple spots corresponds to an immunoglobulin secreting cell. And so we could count that number of cells to see how many made whatever antibody of interest you were looking for. Okay. So there's two ways of measuring antibody production, either through an ELISA or an L-spot, depending on the question you want to address. Okay, so in summary, cell culture is a powerful tool to examine function. There are many different types of media that you can use. It's kind of, if you're just starting out and you don't know, it's a little bit of trial and error to determine the correct media and the different culture conditions. There are different isolation procedures you can use depending on what the question is, on what type of cells you want to look at. There are different functional assays you can be looking at if you want to look at proliferation, if you want to look at cytotoxicity or antibody production. And then you can there's the development of the therapeutic cells, and this is the tumor-infiltrating lymphocytes or the differentiation of the stem cells. Okay, so let's go back to the CME credits then. So which of the following assays could be used to measure proliferation of T cells? CFSC, chromium release, thymidine uptake, A and C, or B and T?
D, A, and C.
Right. Okay, so you can use CFSC and also the thymidine uptake. Which of the following objectives of cell culture does not directly represent a present or planned therapeutic strategy for addressing human disease? A, tumor-infiltrating lymphocytes, differentiation of stem cells, cytotoxicity testing, or monoclonal antibodies from hybridomas. Right. And measurement of antibodies by ELISA is useful as an indicator of immunodeficiency, multiple myeloma, autoimmunity, or D, all of the above. Right. Okay. That's it. Any questions?
You mentioned protein mitogen, which I always thought it was B cells, but I thought it was like you had to have T cells present for the to work or something.
The pokeweed?
It wasn't. I didn't think it was pure B cell. Was it not? Is it? I would think of what one's used for for VMPs, but we would traditionally think of it as B cell. But I thought there was something about that you had to have some T cell help for that to work.
I'm not sure that that's really necessary. I don't know. I don't know that we use pokeweed that much now, because mostly we use Staphylococcus aureus for B cell proliferation.
Because when we send our tests off for proliferation assays, they always use PHA, concanavalin A, and pokeweed mitogen. Those are the three that they use. And those are still used today, very commonly. They don't do Staph aureus. So.
Okay.
I don't know. But is Staph aureus a better one to use?
Well, we know that it's specifically for B cells. Pokeweed, maybe with, well
it's not 100% for B cells, so maybe it's more specific.
Right. And it's interesting they use Con A, because Con A is mostly for mouse cells.
For what?
Mouse lymphocytes.
Mouse lymphocytes. It doesn't work for humans? Because we get good results.
Yeah, but, I mean, traditionally it was used for mouse lymphocytes and PHA, for T cell response.
And then they do tetanus and candida, which give much lower readings because there's a much smaller number of specific lymphocytes. The thing that concerns me is that we still, here we are 25 years since I've been here, and we're still sending mitogen studies off to National Jewish and not doing them here at Children's Mercy. We have a bone marrow transplant unit. We get patients with lots of immunodeficiencies. We're doing lots of advanced things now, and we still don't do our own proliferation studies. Is there a reason for that, or are you at the stage now where maybe you can start to offer those services?
Well, I'm not sure that we're not licensed through, is it, I forgot.
Through the lab.
Is it CLIA, is that what it is?
But you can get licensed.
Yeah.
I mean, you just have to do whatever things that they need you to do, but it just seems to me that the need for proliferation studies is high enough now that we ought to really think about having our own ability to do them.
And I don't see why Dr. Zwick can't offer something like that.
Because he's got other priorities, I guess. It's something that we're going to have to kind of push from our own group.
Our DeGeorge kids. Right. Because right now, I guess at National Jewish, you still get a thymidine.
We still send it out to Jewish, and they do the thymidine.
Yeah.
We don't want to do thymidine because of the radiation.
Right. I mean, you could certainly
But there's all these other things now that can give the same kind of results.
And CSSC will do that for you very well.
You had a comment, Brock?
Yeah, I was just curious about this effector-target ratio. Can one effector kill more than one target?
I don't think so. I think it has to be fairly specific, because it has to recognize it's through its T cell receptor. So depending on what's on that target.
That's fairly specific, but I think the question is, once it's killed that one, can it then go on and kill another one? Or
it can be recycled.
So it can be recycled. Because it looked to me like it covalently binds to the cell, and by killing it, it probably kills itself too. But it doesn't.
No.
It actually can then recharge, go off, and get another cell.
Right. It does.
Oh.
I mean, for example, if you had some cloned cytotoxic T cells to a specific receptor, then that would really change your ratio greatly. I mean, if that occurs.
Right. You're right. That's absolutely right. But with mostly the chromium release assay, it's a heterogeneous population of T cells.
Yeah.
They're doing a cytotoxic T cell assay with.
Right.
All right. Anything else? And we're hearing about these cell culture techniques now that they're growing like whole organs. Obviously, you can't grow a kidney in a flask of culture medium. It seems to me like you'd have to have blood circulating and things like that. So At what point does it become more complicated? How big of a, how
Well,
complex do the cells have to be before more is needed?
I think for an organ culture, we're really talking about fragments of we could probably maybe grow a piece of a lymph node for a little bit in culture, but I don't think we
Once it's more than a certain size.
Yeah.
And there's a certain number of times that these cells can grow. After they multiply a certain number of times, don't they stop growing?
Yeah, unless they're transformed. So if you wanted to establish a B-cell line, then you can transform it with or infect it with Epstein-Barr virus, and then that essentially immortalizes that B-cell, and then you can grow it forever. And with T cells, it's a little more difficult. You can get them growing for a fairly long time as long as you keep them maintained with the feeder cell and antigen and usually IL-2.
Yeah, when I was studying, I used to do cell culture a long time ago, and we were working with this thing called defined medium where you didn't have to use the cap serum. And
okay, right.
Has that gone away now? Have they given up on that, or is that?
No, I think that people still do that you have to replace it. You have to use ferritin, I think, or something like that, with it in the defined. It's part of the defined media. I think that some people still use that, but I don't think all cells will grow in defined media.
So we still don't know what it is in serum that helps these cells to grow. There's still some magic.
Yeah,
Wow.
Whatever is in the serum. And it's much easier for them to grow in the medium with sera than even in the defined, because they have to be adapted to grow in the defined. You just can't easily take them like what we do is just take them out of the lymph node and separate them in the single cell suspension, and they can grow them, and they grow fairly happily. But in defined media, you really have to almost adapt them.
Well, they don't grow as well, but even the serum, when we would ask to get fetal calf serum, and they would put a big supply from the same calf.
Yeah.
If you had a different cap, it wouldn't grow. These cells are really finicky.
Right. Yeah, some cells will do that. You have to have the same lot of fetal blood serum, right? Yeah, that's true.
It's interesting.
Wasn't that called fetuin? The secret substance in fetal calf serum?
It's the special sauce, the secret ingredient. Cell culture people it would become like an alchemy. It would be a magical science.
Right.
And it really became an art.
Yeah, it is an art, very much so. I mean, and you think you're following as closely as possible all the sterile techniques you do, and still yourself will get contaminated. And a lot of it's seasonal as well, because we notice that in the spring we get more fungal contamination. And even though we're in a laminar flow hood and, we've flamed everything is sterile, we'll still get some fungal contamination.
Comes on your clothing, or maybe you're noticing. When you breathe more mold in while you're coming in, now you're breathing it out. It comes and goes. Who knows? It's really interesting. All right. Well, very interesting. Thank you so much.
You're welcome.
Thank you, Marcia.
All right. We're going to take about a 5-minute break, and then we'll be back, and Dr. Miller will tell us about Stevens-Johnson syndrome.
This has been an ACAAI production. To learn more about Conferences on Lyme Allergy or the American College of Allergy, Asthma, and Immunology, go to www.acaai.org. See you next time.